Hi guys, thank you so much for coming over here. Can I get the slides? Okay. Thanks for that wonderful, kind introduction. Look at that flower. Look how beautiful it is, said an artist to his friend. Art appreciates and celebrates that beauty, whereas science just takes that flower apart. Well, the artist was talking to Nobel Prize-winning physicist Richard Feynman, and he thought that that view was a bit nutty. He said, well, as a scientist, I too can appreciate the beauty of that flower. But as a scientist, I can also dig into the details of how the flower gets those colors, the molecular processes inside the cells of a flower to understand why it has that beautiful color to it. And as a scientist, I can think about why the flower has those colors, which is, of course, to attract insects, which then raises interesting questions about how insects perceive colors and how they have evolved to find some aesthetic beauty in that flower. Richard Feynman countered back to the artist that science doesn't take away the beauty of a flower, science only adds. So Richard Feynman said that when I was a young boy. He said it on TV in 1981 with roses swaying behind him in the background. And he said it better than I could at that time, that science really adds to our wonder and appreciation of the world. And I already knew I wanted to be a scientist, but it was that moment that really articulated it better than I could say myself. Now, the human body, at least the insides of the human body, doesn't have an aesthetic beauty in the same way that a flower does. But from the detail, from all our understanding of how the human body works, there is a really profound wonder that lifts us out of our daily life, and we really see something special about who and what we are. The part of the human body which we understand the best, where the details have been worked out the best, is during an immune response. So what happens when you get a cut and there's a red swelling and inflammation and cells move into that cut to deal with any opportunistic infections that might move into that cut? That's the part of the human body that we understand the best. And from all the detailed knowledge that we have about how the immune system works, it's opening up all kinds of new treatments, for example, in autoimmune disease, cancer, and other areas. And so that's what I really want to talk about this morning over the next 50 minutes or so. So I want to spend half the time talking about something about how we got this knowledge. I want to tell some of the stories of the people involved and what they discovered and how they discovered it, and present to you essentially what is a scientific revolution in our understanding of immunity. It's not something that has one particular unique moment. It's something that's been going on for many decades, and I'll present something of that so we can appreciate how we got this new knowledge. Then in the second half of this, I want to talk about what the implications are for our health. I'll present how this new understanding directly leads to treatments for autoimmune disease, cancer, and also look to the future a bit about where new medicines might come, and try to explain why I do think it's not an overhype to suggest that this is a revolution for 21st century medicine. And then if there's time, I'll also touch on some of my own recent research, some of the stuff that's been published very recently. So, any starting place is a bit arbitrary, right? We've been studying the immune system for many decades, if not centuries. But I'm going to begin with Charlie Janeway. So Charlie Janeway, who was a professor at Yale University, he had an epiphany in 1989 that really changed the way we think about how the immune system works. He got to this epiphany by thinking about two particular things that didn't quite make sense, and then he combined them together to have this brilliant idea. Now, the two things he was thinking about, the first thing he was thinking about was how vaccines work. Now, roughly speaking, we all kind of know how a vaccine works, right? So your body is exposed to a harmless or dead version of a germ. The cells in your body that recognize that germ multiply in number, And some of those cells then stay in your body for a long time. So if your body then sees the real version of the germ, you have an immune response that can react quickly to the real germ, and roughly speaking, that's how vaccines work. Everybody at the time that Janeway had this epiphany was working on understanding that in the sense of how is it that the immune cells live in your body for a long time and mount an immune response rapidly the second time round. Now, Charlie Janeway was thinking about how vaccines work, and he realized there was a gap in our understanding of that. And that's because it came from an experiment that was done in 1926, which people weren't really thinking about much at the time, but he did. And the experiment was this. Now, in the way I just described to you about how a vaccine works, it shouldn't matter as to whether you're exposed to the whole version of the dead germ or just a little piece of it. If a body sees a little piece of the germ, you should still be able to mount an immune response to the real germ that has that bit of a germ. But an experiment in 1926 was done where it was done in animals. A mouse was given a dead version of a bacteria or one isolated type of protein molecule from that bacteria. And the mouse would see the dead bacteria. If it was given the real bacteria, it would mount an immune response, and that would be fine. The dead bacteria worked as a vaccine. But a little piece of the bacteria, one type of protein molecule, didn't work as a vaccine. So if the mouse was given a protein molecule from a bacteria, if it saw the actual bacteria, it didn't mount an immune response. An isolated protein molecule could not work to make a vaccine. Now, everyone's thinking about this process of a vaccine in terms of your body reacts to things that are not part of you, right? Your body sees a germ, like a bacteria, and the cells that see that bacteria multiply in number, then they can respond if you see the real bacteria. But if a body reacts against things that are not part of itself, it doesn't make any sense for why the mouse couldn't respond to just an isolated bit of the germ, one type of protein molecule. It didn't work. There's no real reason why that shouldn't work as a vaccine. It got even more bizarre in that just by chance, they put this protein molecule with lots of different chemical situations, and it turned out that if you mixed a protein molecule with an aluminium salt, now it did work as a vaccine. Now the mouse would be able to make an immune response against the actual bacteria if it saw it. Now, Charlie Janeway realized that didn't really make sense. People at the time thought maybe the aluminium salt allowed the protein molecule to live in the animal for a long time, stay in the animal, for a long time, and that would be why it could mount an immune response. But there wasn't really any evidence that was true, and Charlie Janeway realized there was some gap in our understanding. It wasn't that this was some small technical trick which we don't need to worry about. There was a real gap in our understanding of how a vaccine works. It can't be that the body just reacts to anything that's not part of you, because then it should also react to just an isolated protein molecule from a germ, and it doesn't. So that was parked in his mind. He thought there was a really big gap in our understanding of how a vaccine works. He actually called it a dirty little secret. Now, the other thing he was thinking about, he was at a scientific meeting with his wife, who was a fellow immunologist, Kim Bottomley. And he was it was actually a Keystone meeting, so somewhere around here. And all the scientists were presenting their work on how the body mounts an immune response better second time round. Charlie Janeway and his wife suddenly realized, wait a minute, they're all working on this same thing, how the body mounts an immune response better second time round. But what makes the immune reaction start in the first place? Well, it didn't make any sense. We didn't know. They didn't know. What does make an immune reaction start in the first place? No one really understood. So he thought about that for about 6 months, and he then thought about these two things together, that we didn't really understand how an immune reaction starts in the first place, and that we don't quite understand how vaccines work, because there's something about that they only respond to a whole germ, not an isolated bit of a germ. And why is that? And he put these two things together. And his big epiphany was that it can't be that your body reacts to everything that's not part of yourself. It has to react specifically to dangerous germs. okay? It has to react to things that are not part of yourself and are possibly dangerous. And in hindsight, that is quite blatantly clear. You take in food that is not part of yourself, and you don't ideally want your immune system to react against that food. There's obviously things in the air, dust and particles and other things that you don't want your immune system to react against. The body cannot react to things that are just not part of itself. It has to also react specifically to germs. And Charlie Janeway went further, and he said that there has to be a whole part of the immune system which we haven't yet discovered, which has to directly detect the presence of germs. He postulated that on the surface of immune cells would be receptor molecules protruding out that lock on to telltale signs of germs, molecules that would signify that this is a germ because it represents something that a molecule that could only come from a germ. So he stood, oh, my slides have disappeared. Well, he said that he went in 1989 to a meeting in Cold Spring Harbor talking to the world's best immunologists, and he said, look, all of you guys are talking about how the body reacts fast the second time round, but there's a dirty little secret in that. And he said that everyone is studying the immune system as if knowledge is approaching some sort of asymptote, where future experiments are obvious, technically difficult to perform, and aim to achieve ever higher degrees of precision rather than revolutionary changes in our understanding. In other words, he said, you're all missing something big. Everyone has missed how immune reactions start. He said there had to be a second signal needed to kick off an immune reaction, which was a way to directly detect the presence of germs. So he published that in the proceedings of that scientific meeting. And I've asked many people who were present at the meeting, what was that like? Was his epiphany just everyone was like, oh my God, that is amazing? No, they just didn't know. They were like, well, maybe. No one really knew. There was no evidence for what he said. No one really knew if he was just, going off on one or there was some really revolutionary thing that he'd come across. So his paper from that idea was just ignored for 7 years. But it touched one person 4,500 miles away. Ruslan Medvedov was a student in Moscow University, and he was an undergraduate student there. He wasn't allowed in the library that stored Charlie Janeway's paper describing this idea, but Ruslan Medvedov's a lovely, charming guy, and I can well imagine how he charmed his way into the library. He got the paper, and he spent a month's stipend photocopying it. He says it was like a visceral experience. It was like a light bulb moment. This had to make sense. There had to be a way that the immune system directly sees germs. It can't be that the body reacts to just everything that's not part of you. We have to be able to detect germs. So he started to correspond with Charlie Janeway, who's in Yale University. And Ruslan Medvedov, there was one computer. This was the time the Soviet Union was breaking up, and there wasn't a lot of money for scientific work in Moscow University. And Ruslan Medvedov says there was one computer connected to the internet, and they were allowed to send 300 words per day by email. He wasn't allowed to as an undergraduate, but he charmed his way with a librarian. And he wrote out a 300-word email, put it on a floppy disk, gave it to the librarian sent it over to Yale. A message would come back and be put on a floppy disk, given to Ruslan Medvedov, and they would slowly communicate in this way. So Charlie Janeway was really excited. Whoa, now everyone's ignored my idea, which I thought was pretty good. And now this guy in Moscow has taken it up. Well, that's definitely, he was pretty excited about the idea. Eventually, Ruslan Medvedov said, really like to come and work in the US. That'll be wonderful. How about I come and work with you? So Charlie Janeway discussed the idea with his wife, Kim Bottomley, that, well, he's an undergraduate in Moscow. We don't know anything about him. Let's not bother. So they didn't, Ruslan Medvedov didn't get to work with Charlie Janeway for the moment. He got a 3-month fellowship to working in California to look at scanning and aligning and studying gene sequences, which was a new thing to be doing at the time. Soon before he had to move back to the Soviet Union, he happened to give a talk in his broken English, and in the audience was the president then of the American Association for Immunology, Richard Dutton. And Richard Dutton thought that Ruslan Medvedov's talk was pretty good, and afterwards got chatting to him. And he said, oh, yeah, Ruslan, that was a really good talk. what are you doing? He said, well, I've got to go back to Soviet Union soon. But there's this guy in Yale that I'd really like to work with. And so Ruslan Mezitov told Richard Dutton that he was keen to work with Charlie Janeway. Richard Dutton sent an email to Charlie Janeway saying, I've just met this guy, actually I think he's pretty good. And the next morning, Ruslan Mezitov got an email saying he should come over and work with Charlie Janeway at Yale, and finally the two of them met. So now they've got to work on this idea. They've got this, they're all excited about this idea that the body has to have a way to directly detect germs. But they don't have any proof of that. Ruslan Mezitov hasn't done a lot of experimental science before. How are they going to even go about trying to prove this idea? Well, now we're going to shift the story over to Paris. And this is Jules and Danielle Hoffmann. Jules Hoffmann was the head of the lab. Danielle Hoffmann was his PhD student, and they married. Jules Hoffmann, his father was an entomologist. He was interested in insects. And he thought, well, 80% of the world's species are insects. And insects are surely exposed to bacteria, viruses, fungi, just like we are, or any other animal. Now, insects must have some important antimicrobial defense. This does not look like a path to a Nobel Prize in medicine, studying the immune system of insects, but it's going to be. So Jules Hoffmann's lab were trying to work out what is the immune system of an insect. His lab decided to focus on grasshoppers, but half the lab became allergic to grasshoppers, including his wife Danielle. So Jules Hoffmann decided it would be a great idea to move away from grasshoppers and focus on Drosophila fruit flies. And it turned out to be quite important that they did switch to working on these flies. So essentially, the Nobel Prize winning experiment that his lab did was this. They took the flies and they pricked them with a bacteria or a fungi, and then they isolated blood from the fly, and then they showed that the blood from a fly could kill bacteria or fungi in a lab dish. So that meant that the fly had its own strong, inducible immune defense. They proved that the fly has an immune response to these bacteria or fungi. And then the question is, how is this triggered? And it turned out that because they worked on this particular fruit fly, there were lots and lots of other labs working on this particular fly for many other reasons. And there were lots of mutant versions of the fly with their genes changed for studying all sorts of aspects of that fly's behavior. And so they tried different versions of the fly to see if any of them didn't have this particular response, couldn't do this thing of mounting an immune response against a bacteria or a fungi. So they then took genetically modified flies and pricked them with a fungi and found a version of the fly where the blood from that fly couldn't kill off the fungi. So now the immune response in the fly didn't work. And so that identified genes of the fly that were important in its own immune defense, and specifically a gene called the toll gene. So that meant that flies have this particular gene, toll, that was really important for the fly being able to detect the presence of this type of germ. This is the exact data, the graph that they did. These are the flies, and you can see that the mutant flies, the ones lacking that toll gene or having a change in the toll gene to make it not work properly, they were really easily overrun with fungus, whereas normal flies could deal with a fungus infection normally. So that's one version of the data. This is another version of the data. That is the fly dead, overrun with fungus, because it lacks this particular gene that allows it to detect the presence of that fly. So in June 1996, Jules Hoffmann and Charlie Janeway, Ruslan Mezitov, they meet at a scientific meeting, and they discuss all these data, and they tell each other, oh, we've got this gene in a fly that's somehow important for the fly being able to recognize a fungus. And then soon after that, Charlie Janeway and Ruslan Mezitov find a human version of that same gene. Okay? So we have a very similar type of gene like the fly has in us. That's their big discovery. So now there's some issues about whether they got the idea from the insect or they were already working on the idea, but they certainly very soon afterwards came across this monumental discovery that we have some aspect of our immune system very similar to this fly. But we still don't quite yet understand exactly what's going on here. Now we're going to move to Texas, where Bruce Beutler, born in Chicago, is working on a very different, well, seemingly at the time, something very different. And asked people for photos of them at the time they did the work. This is Bruce Beutler, sometime around when he did this monumental discovery. Now, you know, his father was a famous Harvard physician, told him, you've got to work on something really important in your life. And he decided that the thing he was going to work on that was really important was this mouse. Now, many of you will know that sometimes if you have a bacterial infection that sort of goes out of control and the bacteria multiply very rapidly, then it can be damaging, pathologically damaging to the body. Your immune response is so great against the bacteria that you get what's called sepsis. And if that can become life-threatening and called septic shock. And many of you know that can be a huge problem for people. Now, this mouse did not get sepsis or septic shock. So you could give it bacteria and it would never get the problem of sepsis. So that meant to Bruce Beutler and others at the time that the mouse somehow did not really respond properly to bacteria because it's in sepsis or septic shock, it's your immune response itself that causes the problem. And this mouse doesn't get sepsis or septic shock, which meant that the immune response was somehow not seeing the bacteria. So he thought that there was something about that mouse that was really interesting. And if we knew what was wrong with that mouse or different about that mouse, it would give us a clue as to how the mouse is able to see bacteria. So he was looking for a mutation in the genes of the mouse to be able to find out which gene has changed so that it couldn't see bacteria. Now, he'd been looking for this gene for years. He said it was particularly frustrating. It's like looking for a lost coin in your living room. You just never know where it's going to turn up. And also at the time that he actually eventually made a discovery, he was going through a very difficult divorce, and he had other problems in his, many problems in his life. The research body that was funding him to do the work said, you've been doing this for a while, looking for this gene. We're done with it. Your research grants are ended. We're not funding any more of this work. Then, eventually, that's the screenshot from the computer that he made the discovery of, and there was a mutation in a gene that meant that this mouse could not see bacteria. And the mutation was in a gene that was very similar to the gene in the fly, which was similar to the gene in us that was also important. So now things started to make sense. Bruce Beutler saw that this gene is important for how the fly detects bacteria. And once he made the discovery, he phoned back the funding agency, and they said, well, it's too late. We've already cancelled your funding. But in any case, later he won a Nobel Prize. So now we understand really that what Charlie Janeway did predict was true, that there are these genes encode for molecules that protrude out from your immune cells and they directly latch on to bacteria. Interestingly, Bruce Beutler said he didn't really get the idea from Charlie Janeway, that he was coming at it from a different angle. He already knew that mouse was important and then discovered the gene that was mutant in that mouse. So that's something to think about. He says that for Bruce Beutler, it's experiments that drive the work, not thinking about it, not having ideas, and for others, so there are different views as to how science progresses, even within this story that stretches across the globe, across many people. And this is the protein molecule that they discovered. That protrudes. So at the bottom would be where the cell, this part of the molecule would be sticking out from one of your immune cells, and this part here would latch on to this molecule, which is a telltale sign of bacteria. So the molecule that this thing connects with is only present on bacteria. So your immune system uses this to stick to the bacteria, and that tells it that it has definitely seen a bacteria. Now, of course, we've discovered many versions of these types of molecules, and they detect fungi, bacteria, some viruses, and they're all positioned in the body in different places, some of them on cells where they'll be very likely to see fungi or very likely to see bacteria, and there are countless versions of these proteins. Essentially, this opened up a new world of our understanding that your body does have a way to detect what's not part of you, but it also has different ways to latch on very specifically to different kinds of germs, which then instruct your body to react in a different way depending what type of germ they've latched onto. So, Jules Hoffmann says it's important to emphasize the research was all curiosity-driven. He was not seeking anything medical, directly medically important. He was thinking about the immune system of insects. I think the best example or sentence that comes from that, Martin Rees, who was the Astronomer Royal previously, head of the Royal Society in the UK, he says that a research proposal to make flesh appear transparent would not have been funded, and even if it had been, the research surely wouldn't have led to the X-ray. So it is very important that we have some aspects of our research directly targeted against cancer or against specific diseases, but they unravel the whole part of how the immune system works from just really not thinking strongly about a particular type of illness they were setting out to cure. The impact of what they did meant that we understood now that to make a vaccine work, it needs to see something that's not from your body, but also a very specific telltale sign of a germ. And aluminium salt is most likely tricking the body to some extent, making it think that it's seen something from a germ. It triggers some signaling inside a cell to make it seem like it's seen a germ. And then that led, at the time, pharmaceutical companies weren't that bothered about vaccines because there wasn't an easy way to make lots of money from them. But now we understood that you need to trigger this part of the immune system. They did get interested in vaccines. And one of the very specific things that came out from this kind of work is, for example, the human papillomavirus vaccine. It also changed the mission of the immune system. Up until this point, everyone was thinking your immune system works to attack what's not part of you. They changed that. It's about attacking what's not part of you and is from germs. It also brought about our understanding of the complexity of immunity, that you have all these different types of, it's not a simple system. There's lots of different types of cells, some very good at detecting bacteria, some seeing viruses, some seeing fungi, and they have to all interact together, and they all together amount to the immune system that we have. It's not just one system, it's a really complex network of many different genes, proteins, and cells, that many different subsystems that interact together to give you this complexity that is the immune system that we have inside us. This is a quick summary of everything I've said. So just take that in. So you have these different protruding proteins that detect different germs. They send off these events inside our cells. These different protein molecules bind other different protein molecules, and they trigger a response appropriate to the germ. Each arrow here is 3 or 4 years' work by one person. So although I've highlighted the fact that, people made huge leaps forward in our understanding, this is the work of tens of thousands of people across the globe in a community of immunologists studying how the system works. This is another summary of everything I just said. This is one of your white blood cells moving around chasing a bacteria. Everything I've just told you is guiding this process of how this immune cell knows that that is a germ it needs to get. There are red blood cells that it's pushing out of the way, and eventually it gets it. Okay, so now I want to talk about the implications of how this leads to new medicines. So we have this revolution in thinking about the immune system, the complexity of it all, and one of the things that comes from that is that the cells have to interact with each other. The immune cells have to talk to each other to mount an effective immune response. And one of the ways that they do that is that they send soluble secretions out to each other to inform each other, to get each other excited about a particular type of germ that's present, for example. This is Tiny Mania and Mark Feldman. Mark Feldman was on holiday in the Costa Brava of Spain. He actually, my lab is in the UK, so I don't know where the film was going, but never mind. I'll tell you guys, they won't hear. Mark Feldman always tells me you need to tell your lab team to take very long holidays. Okay, keep that to yourself. So, because he was on holiday and he had a brilliant idea. Sitting in near some 15th century castle ruin on the Costa Brava coast of Spain, he thought, well, in autoimmune disease, we know that that's caused by your immune response reacting to something that it shouldn't, some part of yourself, and by mistake, that's causing a problem and leading to the symptoms of a disease. And if the immune cells have to talk to each other to mount an immune response by these soluble secretions, then perhaps blocking the soluble secretions that immune cells send out to each other, we could stop autoimmune disease. So he published just the idea in The Lancet. Nowadays, it'll be quite hard, I think, to publish just an idea in something as prestigious as The Lancet without really an experimental proof of that. But he published the idea in The Lancet. And then he got to work with Tiny Mady. Tiny Mady is, by the way, born in Ludhiana in India. Mark Feldman is born in Poland. And actually, Mark Feldman himself would say that it's really important to emphasize that immigrants did this work. They met in London, and Mark Feldman's Tiny Many had this idea we should look at autoimmune disease. Tiny Many thought we should look in rheumatoid arthritis because where the problem is relatively easy to access. In things like multiple sclerosis, the human tissue is hard to access, whereas synovial fluid from a knee or a joint is relatively easy to access. So they looked at what soluble secretions are present in rheumatoid arthritis patients in their joints. They found a particular molecule coming out of immune cells. It's called TNF. It doesn't really matter what the name of it is. And then they decided they could block that soluble secretion. So it would stop immune cells from egging each other on to mount an immune response. And many of you will know that that is a multi-billion-dollar drug, whether the money is important or not, who cares. But the fact is that because they blocked those secretions, it helps many, many thousands of people. And many people are not in a wheelchair because of that particular drug that stops the secretions coming out from one cell to another. I'm conscious of the time, so I'm running out of time for all I want to tell you. So I can't really go into this story so much, but the person who actually made the reagent that blocks those secretions is Jan Wilczek. His story is also one it's truly inspiring. He was born in Czechoslovakia to escape persecution when he was young. His parents took him to live in a Catholic nunnery. That became unsafe. His mum went to live with him in a village. That became unsafe. They lived to an even more remote village. Later, after the Holocaust, he then set up his lab in Czechoslovakia. Genes were not allowed to be spoken about because of Levchensky's ideas about how inheritance works. So eventually he defected over to New York, and he then made the version of the antibody that would block these secretions, the TNF antibody, and this is him getting a medal from Barack Obama, and he told me that the one thing he really wished that would have happened with this was that his parents would have lived to hear about that. And in fact, he also, from the money he made, relatively recently, he donated the largest amount of money ever to a hospital, which was about $100 million to a hospital in New York. I asked him about it, he said, well, no one should come to New York to live unless he's willing to be lucky. Okay, this is Jim Allison and his wife Padmini Sharma. Now we're going to talk about cancer. And this is Tasuka Honju in Japan, in Kyoto University, shortly after he won but both of these people, Jim Allison and Tasuka Honju, won the Nobel Prize fairly recently. Many of you will know. And they won the Nobel Prize for a new type of cancer therapy. Jim Allison was not focused on working on cancer. He was studying how the immune system works. And essentially, from curiosity, there was a particular protein molecule, a gene that encoded a protein molecule that looked similar to another type of gene, and he was just investigating how this works. Not just himself, others as well, Jeff Bluestone and some other laboratories. And they discovered what's now known as the immune system breaks. And at the time, it was quite a revolutionary discovery. But in hindsight, it makes a lot of sense. When you're infected with the flu virus, for example, then as we've said, in the context of a vaccine, we talked about it, the immune cells that see that flu virus will multiply in number to kill off the flu. But when your body has cleared away the flu virus, you don't want all these immune cells there just fighting off the flu. So your body has to come back to its normal resting state. So that means that the immune cells have breaks within them to switch off the immune response after some time. So when your body has eradicated the flu virus, then you switch off that part of your immune system to bring it back to its normal resting level. And Jim Allison in particular realized that if your body is switching off the brakes in another situation where there's a long-term problem in the body, then that might not be good news. So for example, in cancer, you don't want your immune system to switch off its response after some time. So he decided that maybe blocking the brakes on the immune system, switching off the brakes, might be good in some situations of cancer. In itself, the body reacting to cancer is quite a profound thing, because obviously cancer is just your own cells that have gone awry. They've started to multiply out of control. That means there's nothing obviously different about them in the same way that a virus or a bacteria is very different. And so for a long time, we didn't even think your immune system could mount a response against cancer. But we discovered over time, many other scientists involved in this discovered that the mutations that happen to make a cell cancerous can be picked up by the immune system. And that means that if the brakes then come on that part of the immune system, that might be detrimental to how good your body is at fighting off cancer. So essentially, your immune cells can see tumor cells, they can react and kill off cancerous cells, but after some time, if the brakes might come on, tumors might specifically exploit this and put up receptors that trigger brakes on the immune response to switch it off. And so he decided, in fact, not entirely dissimilar to the ideas of Mark Feldman and Tiny Many in stopping cells communicate with each other, they would use the similar reagent and antibody to block the brakes on the immune system. Now, that works for some cancer patients. We don't want to overhype it. That works for some cancer patients with particular types of cancer. But what was really exciting about that was that some patients that were having a type of cancer where there was really normally only a very short time for them to survive, that for some lucky patients, there would be a really long-term ability for them to survive their cancer. I mean, it's hard to use the C word, cure, but they certainly live for a very long time after receiving this kind of treatment. But it does only work for a small number of patients, relatively around 20% in the best situations with some types of cancer. But I'm not here to fool you and tell you that this is it, this is how we're solving cancer. That's not right. But what is right, what's not an overhype, is to really suggest that this is the tip of the iceberg. There are currently, I think there's 6 licensed versions of these antibodies to treat types of cancer, and essentially they block 2 specific brake receptors on the surface of immune cells. But altogether, there are about 20 different brakes that immune cells use. And each of those brakes switch off different parts of the immune system or switch them off at different times or in different places in the body. And so the reason that this is exciting, why this is not an overhype to say this tip of the iceberg, is that it opens up the ideas of which other receptors and brakes we can switch on. So that there are countless ways in which we might be able to tweak the power of the immune system, to boost its ability to fight off cancer or to dampen the problems it's causing during autoimmune disease. We can try switching on each of these other breaks, the other 20 breaks, and we can block them in combination with each other or in combination with other drugs. There's almost endless numbers of different things we could try and do, and we don't have enough knowledge yet to really know what's going to work. So the way we're doing that, essentially across the globe in small startup companies and in large pharma and in academic labs, between us all, we're trying all the possible ideas. And some of them are going to work. And that's why it's an exciting revolutionary time for how we will tweak the power of our immune system to deal with different kinds of diseases. Jim Allison, they become, is well known also, becomes a kind of celebrity status for this wonderful work that he did. This is him playing in the House of Blues. Here he is meeting Willie Nelson, playing in the House of Blues. Here is this Sean Parker, who's donated lots of money to pursue these immune therapies. Here he is with the Edge from U2. But it's the next photo that him and the whole community of immunologists are proud of. This is Jim Allison with Sharon Belvin. Sharon Belvin was given 6 months to live, and because of the antibody drug that Jim Allison made, Sharon Belvin is still alive well more than 10 years since she had that short time to live. They kept in touch. Sharon Belvin told Jim Allison, when she had her first child, a couple of years later, she told him when she had her second child. And both the Quran and the Talmud say that anyone who's saved one person's life has saved the whole world. Seem to have... My slides have gone off. Okay, sorry. Okay, I only have a few minutes left, unfortunately. Let me just tell you a little bit about my own research. My PhD is in physics. I came to the US to study immunology at Harvard. This is the type of work that I did to visualize using new high-powered microscopes how an immune cell detects signs of disease in other cells. The sort of movie I showed you, of it rushing about to chase a bacteria, I want to see that process in very high special resolution to see exactly how that process works. I discovered something called the immune synapse, which is that at the contact an immune cell makes with another cell, the particular protein molecules that are important in that decision. So the same way that we showed you that proteins would see a telltale sign of a germ or they communicate with each other, they don't accumulate at the contacts between cells in an ordinary way. They organize themselves as a specific pattern. And that's reminiscent of what was known to happen between neurons. And that then led to this being called the immune synapse. It's similar to how neurons interact with each other. But of course, immune cells make very transient contacts with each other to communicate, whereas neurons have very long-lived contacts. One of the things that neurons famously do is they communicate with each other over long, thin strands of membrane called axons. And immune cells aren't thought to do that. They're thought to communicate with each other with these soluble secretions, the things we mentioned that you could block in autoimmune disease. Immune cells weren't thought to interact with long, thin strands of membrane like neurons. But in fact, with our microscopes, we saw that they did. And these are long, thin strands of membrane that keep immune cells connected, and we named them membrane nanotubes. Those nanotubes can allow a cell to kill a distant tumor cell, at least in a lab dish. Whether or not that happens in the body is still hard to know. But germs can also exploit these connections. And this is a cell infected with the HIV virus, colored in green, and then the protein in that virus that's colored green moves along the nanotube to infect a previously uninfected cell, showing that germs could exploit these strands of membrane between each other. That's going to present to you a little bit of couple of slides from something we published very recently, just to give you a sense of how this is a never-ending, wonderful endeavor from all kinds of people. This is one of the things that happens in the immune response is when your immune cells detect say a cancer cell or some other disease cell, is that the receptors, the proteins at the surface that see that sign of disease, they often get chopped off from the surface. And so people thought that these receptors that get chopped off would very often, must be part of the immune system to switch off itself. We spoke about how the immune system has to switch itself off after some time. Turns out drug companies were trying to find ways to stop those receptors getting chopped off so that you would boost your immune system to make it fight off better. And it turns out that that might well be completely wrong. That in fact, the chopping off the receptors surprisingly, can boost your immune response, not make it less strong. And the reason for that is that when an immune cell sticks to another cell, which might be a cancer cell, then it would see that that's a cancer cell and it would kill it, then in fact it has to chop off those receptors that saw that that cell was diseased so that it could detach from that cell and then go and kill another cell. So that's completely the opposite of what people thought, and the regulation of how an immune cell detaches from one cell and goes to kill another cell is another aspect of the immune system which may or may not be amenable to new kinds of drugs in the future. So that's just an example from my own lab, but it's similar to many other, how the kind of work that we're doing that digs into details about how the immune system works, which gives us new ideas for medicines. So I've told you about the revolution in immunity. I tried to explain something about how we got there. A huge endeavor of tens of thousands of people across the globe brought us to this point where we have a detailed knowledge of how the immune system works. There are mysteries remain, not everything's understood, but from the knowledge we have, we can get new medicines. I wanted to explain to you that we get autoimmune, how we can open up new ideas for treating autoimmune disease, how it led to treatments for cancer, something about the future, looking at an example from my own lab in sequential killing. Do I have 2 minutes? Yeah. I want to read to you something else because I want to say something else to end on. Science is many things. A method, a journey, a route to power, a body of knowledge, a thing you loved or hated at school, a jigsaw puzzle with an infinite number of pieces, a force for good and bad which has produced both food and bombs. Arguably, its greatest successes has been and will be for some time to come in curing diseases. Still, our own body's remedy, the immune system, is far more powerful than any medicine we have devised. Most germs are tackled by our body without us hardly knowing. Over many decades, we've begun to unpick how this works by testing what happens when one type of cell is missing or in abundance, when a gene is inactivated or enhanced, when a chemical pathway is switched on or off. Step by step, with an occasional misstep, we've uncovered many of our immune system secrets. But like the solar system, the financial system, any big system, the immune system does remain enigmatic. All the theories have cracks in them. Each idea only works in certain situations. Everything is only some kind of an approximation. I and thousands of others continue to devote our days and lives to solving the puzzles that remain. A few principles that capture precisely how it all works, or one diagram that fits on a t-shirt. But that dream may never work out. And it might even be the wrong thing to aim for. In the near future, irrespective of whether or not we can describe the system in simple terms, we may be able to make predictions about one's health by taking few precise measurements, a detailed analysis of different blood cells, say, and plugging that information into a mathematical or computational simulation, just in the same way that mathematics provides the only way to describe how light behaves. In the meantime, using the knowledge we have now, we can make smart lifestyle choices and we create new medicines to fight infections, cancer, autoimmune disease, and other maladies. But death will not be eradicated. Paradise is not promised. From here, we must step forward carefully, thinking deeply about what we set out to treat. Not all deaf people want to join the hearing world. And how shocking it feels now to know that in the UK in the 1950s and 60s, homosexuality was seen as a condition to be treated with the female hormone estrogen or electric shocks. Science is many things, but there are some things it must not be. This must not be the pursuit of any supposed perfection in the human body. So, I've tried to tell you, where this has got us, that we've got a detailed understanding. It leads to new medicines. It also throws up so many new questions I think are really important. We're at the point where we really have to think carefully about the things we really have to treat. I have to say that it's working towards new medicines that drives many of the people involved in the endeavor. But for me personally, it's not actually really about that. It's really because, as I said in the beginning, when Richard Feynman talked about the flower, that all of this opens up a hidden wonder in what we really are. That all of this, the science and the journey, means that we see ourselves for what we truly are, which lifts us out of the humdrum. This is our escape by ascent. Thanks for listening.